Hi everyone, it's Dr. B here, and this is Physical Principles of Quantum Biology, Chapter 5, Light Receptors, Spin Chemistry, and Cryptochrome. I'm Nathan Babcock, and this is work I've completed with my wife, Brandy Babcock, on our book, Physical Principles of Quantum Biology. You can download your copy from the archive at the link shown here. This technical monograph provides a comprehensive overview of the field of quantum biology. In today's lecture, we'll be discussing Chapter 5 on light receptors, spin chemistry, and cryptochrome. So, let's begin. Biological photoreceptors, that is, photoreceptor proteins, are important in vision systems and many other aspects of physiology. During vision, of course, there's a sophisticated photon detection system that enables complex visual processing and spatial navigations. Proteins, like the one shown here, rhodopsin, are critical for this process. It allows us to see. Now, photoreceptors also play a role in other processes, such as circadian control, which is a light-driven entrainment of biological rhythms that govern sleep, metabolism, and physiology. The protein cryptochrome is known to play a role in circadian rhythm regulation, and it's also been hypothesized to play a significant role in magnetoreception. That is, the sense of the Earth's magnetic field that's used for spatial navigation and orientation in a number of different species. Now, plant morphogenesis also has a light-regulated component, and developmental programs to control growth patterns and morphology can be influenced by proteins like phytochrome. If anyone's had a potted plant in your house, in the audience, you may have noticed it tends to grow towards the light. So at the molecular scale, quantum electrodynamics govern light absorption, electronic excitations, and some charge transfer processes via Fermi's golden rule. Now, there are many photopigments such as flavins, retinoids, bilins, and porphyrins, carotenoids, melanins, and other aromatic molecules like the amino acid tryptophan that can also have photoactive roles in biology. During visual transduction, that is the process of transforming light signals, visual signals in the eye, into that neurological impulse that's processed in the brain. The protein rhodopsin acts as the photoreceptor. Rhodopsin shown here in the figure, and if you look within the protein structure, the ribbon structure there, you can see the light active pigment flanked by other aromatic amino acids. That pigment, retinol, we'll talk about in a moment, but for now, let's just talk about opsins. These are proteins like rhodopsin. They're G protein coupled receptors, GPCRs, 7 transmembrane helices, as you can see in the figure. The retinal chromophore forms a protonated shift base with the amino acid lysine, which redshifts the absorption spectrum from the ultraviolet into visible wavelengths. Opsins have exceptional light sensitivity. Rod photoreceptors achieve near single photon detection capability, and cone cells provide color discrimination in the eye. Both systems demonstrate comparable photoefficiencies and active lifetimes optimized for their specific functions. Now, it's also worth noting at this point that many olfactory receptors, these are molecules that enable our sense of smell, are also G protein coupled receptors. Now, olfaction is believed to rely on the formation of ligand receptor hydrogen bonding networks, where the odorants provide ligands that serve as electron donors and/or acceptors in an electron tunneling network, implicating quantum biology as well in this process. Now, high-resolution structural models of olfactory receptors have been experimentally difficult to obtain, so not as much is known, but it's worth mentioning that they are likely to have similar structure and even some similar functional aspects to light receptive proteins like rhodopsin. If we look at retinol, rhodopsin's photopigment, it has two different forms. The 13-cis retinol, which you can see here beside the all-trans retinol. And so when photoactivation occurs, that is, the receptor absorbs a photon of light, photoisomerization occurs, you can see that even though the two molecules shown in the panel here are the same, one is a little twisted up at the end. The cis-retinol has a slightly different shape, a little bit of a kind of a J hook in it. And when it absorbs or emits light, it can undergo a transformation between these two conformational states, straightening out into the all-trans retinol, for example. And so this process releases energy into the photoreceptor protein as the little pigment buried inside those 7 alpha helices you saw on the previous slide. As it releases energy into the protein, there is now a conformational change, not just in the pigment itself, as you can see here, but in the protein structure encapsulating it. Isomerization drives a protein conformational change, activating the signaling state through coordinated structural rearrangements in step 2. Now that leads to step 3, phototransduction and signal amplification. The activated rhodopsin, known as metarhodopsin 2, can in turn activate hundreds of transducin receptors, inducing ion channel closure and cell polarization, and ultimately producing that neuronal signal that gets to the brain and the optic nerve into the optic processing center of the brain, where optical and awareness of vision occurs. Now there's another opsin, melanopsin. Similar to some other proteins I mentioned, the olfactory receptors. In fact, the structure of melanopsin is not well known, but here's an AI-generated structure that was produced by the AlphaFold project that gives you a sense of what melanopsin might look like, the structure it might have. Now, melanopsin's discovery was significant because it expanded the opsin family beyond the classical visual photoreceptors, beyond protein light receptors that we use to see. Because in fact, melanopsin, even though it's an opsin, is not used in vision. And its discovery revealed that there are also widespread non-visual light detecting systems in the body, although this might not be surprising. The physiological function of melanopsin is to regulate circadian rhythm, circadian entrainment, sleep-wake cycles, pupil dilation reflexes, and other broader homeostatic processes in many tissues, not just the eye. It's found throughout the body. And the photochemical mechanism, nevertheless, is much the same. It uses identical retinal photoisomerization chemistry as the visual opsins, but serves these other distinct non-visual neural pathways both in the eye and in other tissues. So, the interesting thing is the discovery of melanopsin really heralded the discovery and isolation of cryptochrome. Cryptochromes are blue light photoreceptors found in plants, animals, and other microbes. They're extremely versatile proteins, and they regulate growth, morphogenesis, phototaxis, and of course, circadian timing as well. You can see an image of cryptochrome in the panel here. The historical precedent for the discovery of cryptochrome, it began in the 19th century. In fact, the plant recognition of blue light has been known for many, many years, into not last century, but the previous century, and light-activated flavoproteins were anticipated as early as 1950. But the pigments responsible for plant developmental responses to near UV and blue light spectra were not characterized until the 1970s, when they were dubbed cryptochromes. So this circadian photoreceptor was theorized because research on mice that were deficient in vitamin A, remember that's retinol, a chemical precursor for retinol, the pigment in opsins, it prompted the hypothesis that there was another class of pigments that might play an important regulatory role in circadian light entrainment in mammals. The experimenters bred these vitamin A deficient mice, and they found that they could still regulate their circadian rhythms even though they could not produce the pigment needed for opsins, for melanopsin. And so this led to the hypothesis that there must be another photoreceptor involved in circadian entrainment that didn't require vitamin A, didn't require retinol type pigment. And in fact, the discovery and isolation of cryptochrome came later. Blue light responses were first mapped to the Ch.5 gene in the rhodopsin thaliana in the 1980s. And by 1993, Margaret Ahmad and her colleague Cashmore were able to isolate the protein responsible and assigned it the name cryptochrome. The name cryptochrome had previously been proposed for the name of pigment in, well, to be honest, cryptogamic plants. And although it was not able to be isolated at that time, scientists were well aware of it, and thus it was given the name cryptochrome, both for its mysterious nature and, as I mentioned, its frequent presence in cryptogamic plants that were being studied at the time. So it was quickly realized that cryptochrome has a homology, that's a structural similarity, to photolyase. The flavin adenine dinucleotide pigment was identified as its blue light receptor molecule, and by establishing homology with DNA photolyases, cryptochromes were then basically, set on the stage as not DNA repair enzymes like photolyses, which we'll get to, but as circadian clock regulators like melanopsin. And, well, that's the story of cryptochrome. Circadian biology is related to magnetobiology, the study of magnetic fields on living systems. Now, cryptochromes have an important role in the core circadian function as they serve as the primary circadian photoreceptors across diverse species, and disruption in circadian rhythms are linked to all kinds of problems, neuropsychiatric disorders, metabolic health disorders, and so forth. Maintaining circadian rhythms is very important to regulation of life. Now, what's interesting is that electromagnetic fields can apparently modulate their function. Low-intensity electromagnetic fields have been found to alter circadian markers and, in some cases, improve sleep-wake patterns in controlled animal studies. Now, this finding led to the proposal that cryptochrome itself could be a magnetoreceptor as well as a light receptor. Cryptochrome's central role in circadian circuits positions it as a candidate for mediating electromagnetic effects on physiology more broadly. Cryptochrome then came to be proposed as the magnetoreceptor used by birds. Behavioral evidence began to accrue. Now, it goes back quite a long ways as well. Avian magnetoreception was documented at least since the 1800s, but the first controlled experiments on birds to test their magnetic sense weren't performed until the 1960s. Now, following on that, a quantum chemical compass was proposed as the visual compass systems that birds use to navigate during migrations, for example, with the help of magnetic fields, along with, of course, other environmental cues. This visual compass system is light initiated. And it implicates a blue-green photoreceptor that is sensitive to Earth-strength magnetic fields. So it was proposed that radical pairs that are formed in cryptochrome, these are electron pairs that are separated so they can undergo spin dynamics. I'll say more about that in a moment, but typically in a molecular bond, for example, a pair of electrons are bonded together so that they share a spin zero state. Their angular momentum between the two is equal and opposite and cancels out. But if you excite one of those electrons so that the pair of electrons previously bonded together now can move some distance apart, this can create what's known as a radical pair. And cryptochrome molecules have a fairly unique capability because of that FAD, flavin adenine dinucleotide cofactor, to generate photoinduced and thus magnetosensitive radical pairs through electron transfer that is initiated by the light absorption step. Now, it's a very interesting area of research because the interaction of the Earth's magnetic field with a single electron is a very weak interaction. The thermal energy, the ambient, energy that is available in the system simply due to the presence of heat, heating processes, is, physicists, we think of energy in terms of kT. That's Boltzmann's constant K times the temperature, and that can give us an energy in joules. It's about 4.1 times 10 to the power of negative 21 joules at ambient temperatures, about 300 Kelvin. Now, the Earth's field interacting with an electron, if you look at the equation that describes that, what's called the Bohr magneton multiplied by the ambient field strength, B, this is about 4.6 times 10 to the negative 28 joules for a magnetic field of about half a Gauss. That's the strength of the Earth's magnetic field. Now, you can see, even though these numbers are superficially similar, actually the strength of the interaction between the Earth's magnetic field and a single electron is 7 orders of magnitude smaller than the thermal energy. This is many, many, many times smaller than the thermal energy. So it was initially hard to imagine how such a tiny, minuscule energy and interaction strength could have any effect on biology at all when the ambient thermal energy is already so much larger. So this presented, if you follow me, the thermodynamic challenge of explaining how the Earth-strength magnetic interactions per electron could have any influence on biology at all. But the key aspect of the theory, this quantum mechanical theory based on this so-called radical pair mechanism, is that the reaction is highly selective. So quantum mechanical selection rules enable an otherwise spin-forbidden biochemical reaction. Is that if the photon of light hits the cryptochrome to produce this excited electronic state, the radical pair state, it's prepared in a particular quantum state, usually a singlet state. That's the same state that the electrons shared in the bond. But the thing is that in the presence of a weak magnetic field, an otherwise forbidden transition from the electronic state of the radical pair from a singlet quantum state to a triplet quantum state now becomes allowed. So even with very large ambient thermal noise present, this reaction can't occur without the presence of a weak magnetic field in order to create the symmetry breaking reaction, in order to make this forbidden reaction allowed. And this is crucial in order to understand the influence of the radical pair mechanism. It's not as critical that there's all this excess thermal energy around, because no amount of energy could make the reaction occur without the presence of the magnetic field, even a very weak magnetic field like that of the Earth. Now, the photochemical dynamics are significant because, of course, when a photon of light is absorbed by cryptochrome, blue or UV frequency light, it carries a lot more energy than the ambient thermal energy. And now your usual assumptions that this reaction would simply be drowned out by the thermal noise no longer applies, because the system is no longer in equilibrium with the surrounding thermal bath. Now, this gives a weak signal the possibility of exceeding the thermodynamic threshold. So it's still the question of how such a weak signal is ultimately amplified, even given this additional non-equilibrium advantage, is still not clear. But the coherent dynamics that enable the transition from the singlet state to the triplet state likely involve a nonlinear mechanism, and perhaps, if you read the book, we say more about this, an effect like stochastic resonance. It's a well-known effect in biology and other areas of physics and engineering research. Stochastic resonance is the process by which a weak signal, much smaller than an ambient noise of the system, can be amplified above the ambient noise level. And so this combination of spin selectivity, singlet, triplet, spin dynamics, non-equilibrium photoexcitation, and nonlinear amplification mechanism, such as a stochastic resonance mechanism, can all come together to explain how birds can have this visual sense of the Earth magnetic field. Visual because it has to be like activated, and in fact it has been shown that the sense itself is operates through the bird's its vision system. So, fundamentals of spin chemistry, this radical pair mechanism. How does this really work? I described it a bit on the previous slide, but you can see this figure here from one of my papers. It shows a chain of tryptophan residues and the flavin FAD cofactor. So when light is absorbed by the system, an electron can be transferred down, or in this case kind of up and to the left, along the chain of tryptophans through the FAD, which has absorbed a photon and gone into an excited state, creating room for one more electron in the ground state. Now, the result of this is to form a pair of electrons distantly separated that have opposite spins, but are not in a single bond between the FAD and the tryptophan, at the end of the chain. Now, this is important because it mediates the separation of the radical pair that allows for significant singlet-triplet dynamics to occur. And in the presence of an additional spin particle, perhaps a nuclear spin that can be found in a number of different atoms in the FAD, or an additional spin in the radical bystander, or so forth. The exact details of the mechanism are not known, but ultimately, in the presence of an additional dipole, an additional spin particle, this radical pair mechanism can produce significant magnetic effect on the reaction. What reaction are we talking about? Well, it's really a question of now, once this excited state with the electrons separated has been created, whether the two electrons ultimately recombine and go back to their original unexcited state, or whether the excited electron escapes, producing a potential signal and possible conformational rearrangement in the cryptochrome to enable higher level signaling to occur. Now, the key signatures of radical pair mediated magnetically sensitive reaction, electron transfer reaction of this kind, include, of course, light sensitivity, directional anisotropy. It depends on the direction of the magnetic field. It's, however, insensitive to the polarity of the magnetic field. So the angle the field makes is important, but you can flip the north-south axis, so to speak, and it would produce the same result. It's also vulnerable to disruption by radio frequencies, which came to be important in the experiments, of course, that tested the hypothesis that if, in fact, the avian magnetic sense is mediated by the radical pair mechanism, that it would be sensitive to radio frequency noise. So, if we want to model this kind of reaction, theoretically, the traditional approach is the so-called Haber-Korn-Master equation, which describes coherent spin dynamics with decay and reaction channels using standard kinetic frameworks. I described escape channels due to losses versus the probability that the radical pair simply recombines. Now, more advanced mathematical methods have also been proposed involving, again, open quantum systems approaches, similar to the Haber-Korn type equation, but they involve non-Markovian effects. Now we assume that the bath has some memory attached to it, that there's some significant interaction between the thermal environment surrounding the radical pair, and it has an alternative reaction superoperator in the mathematical description to provide more realistic environmental coupling. Now, critical interactions, as I mentioned before, include this nuclear interaction, hyperfine interaction between the radical pair electrons and any nearby nuclei in the molecules themselves. Possible spin-orbit effects, although these would typically be negligible. Dipolar interactions to other radicals or spin-containing particles that the radical pair can interact with. And exchange mechanisms. These are quantum fermionic exchange mechanisms that are significant even between the original two radicals themselves. Now, orientation-dependent effects determine the ultimate compass sensitivity and its directional resolution. If we look at this in a little more detail, in a molecule like cryptochrome, as I mentioned, the traditional approach is to use the Haber-Korn-Master equation that's shown below. It's a density matrix equation under a Markovian approximation that assumes that the bath has no memory. Anything that goes out into the thermal environment is just lost, completely forgotten, so to speak. And we have what's called this master equation approach, where we have the time derivative of the density matrix of this system. And it has a first component that describes the coherent unitary time evolution of the system in the presence of whatever Hamiltonian, whatever mathematical equation describes its dynamics, as well as a loss term that describes the probability that, well, ultimately probabilities that the electron either recombines or escapes. Now, advanced methods can include other environmental couplings, other memory effects in the thermal bath, and orientation-dependent anisotropies that can also be important to compass sensitivity. And a lot of work have been done to expand the traditional approach to include other more realistic effects. But ultimately, again, the key interactions are these nuclear hyperfine interactions with the electrons, other electron-electron dipolar interactions coupling to other possible electrons, spin exchange effects, and even in some theories, the possibility of spin-orbit coupling effects. Now, let's talk about the evidence. The radical pair mechanism allows us to make predictions that can be tested against physiological experiments using birds or other species like fruit flies. So, as I mentioned, radio frequency fields are proposed to disrupt the radical pair mechanism. Weak RF fields in the 0.1 to 10 megahertz range do in fact disrupt avian orientation behavior under controlled laboratory conditions, and this supports the idea that the radical pair mechanism is involved in magnetoreception. There's also in vitro evidence performed on cryptochrome in a photochemistry laboratory that shows that cryptochrome exhibits magnetic chemistry, magnetic field effects on its photochemical electron transfer reaction at millitesla and even sub-millitesla field strengths. Similarly, the molecule photolyase has demonstrated comparable magnetic responses. And so model synthetic compass systems have also been tested. These are not proteins like cryptochrome or photolyase, but other systems that can be used to photogenerate radical pairs. And these synthetic chemical compass systems do, in fact, have been shown to respond to Earth-strength magnetic fields under the right photoactivated conditions, proving the essential viability of the theory, the idea that a single molecule can be used as a photoactive receptor in order to detect the strength and direction of a magnetic field of comparable strength to the Earth. Now, cross-species evidence, not just birds, but other cryptochrome-dependent magnetosensitive effects have been identified in Drosophila, that's fruit flies, cockroaches, and even plant systems, alongside the extensive animal behavioral studies. Although some of these results are still debated, and the plant systems, of course, they don't navigate. They just happen to have flavins in them, and so they can, under highly controlled circumstances, demonstrate magnetosensitive responses to blue light as well. Now, the challenge and the conceptual refinements that may need to accompany these challenges to the radical pair model of avian magnetoreception are great. First of all, there are issues with anisotropy suppression, that there are other interactions, dipole-dipole, and exchange interactions, even between the two electrons in the radical pair itself, that can suppress compass anisotropy. That's the compass's sensitivity to the direction of the magnetic field under physiologic realistic conditions. So this challenges the essential viability of the compass system because if you fully include all these different interactions, sometimes the magnetic effect, even in your theoretical modeling, just goes away. And so, and this is some of my work, other theories have been proposed involving other dipole interactions or radical scavenging reactions that can, somewhat surprisingly, enhance the anisotropic magnetic field effect. And this could be used potentially to recover lost sensitivity due to, for example, exchange interaction effects through the amplification that can be generated through dipole coupling or radical scavenging. That's another molecule comes around and reacts with the radical pair before it can finish its initial reaction one way or the other. Now, the frequency sensitivity also has some open questions. Weak broadband RF noise is sometimes more destructive than strong narrowband signals, which might be unexpected. And low frequency noise effects across experiments have been inconsistent. And so it is challenging to produce a consistent body of evidence to show exactly which frequencies of noise are most disruptive and why, and to some extent how, wideband or narrowband, and so forth. Of course, the radical pairs themselves are in question because it's hard to really, get into the animal's eye and see what's happening during the actual visual sense of the magnetic field that the bird, for example, exhibits. And so the canonical radical pair is a flavin, that's an FAD, and a tryptophan radical pair, but also the FAD, that's a flavin and a superoxide molecule, have also been proposed as radical pairs. A flavin and an ascorbate, that's a vitamin C molecule, and even flavin diradical reaction has been proposed, where the individual flavin adenine dinucleotide molecule carries both radicals after the excitation process in order to produce a magnetic field effect, although the fact that the electrons stay very close together make it challenging to consider how this could act as a compass as well, because the distance separation between the electrons is also important based on theoretical modeling to the effect. Now, there's a lot of structural complexity in this system as well that can lead to functional changes. A very interesting result concerns the cryptochrome C-terminal tail. It seems that the intrinsically disordered region at the end of the cryptochrome molecule is the only part that's really essential for circadian signaling and ultimately neuronal magnetic sensitivity. And so if we take a look at cryptochrome, here it is, and you can see I've highlighted in another color, magenta, the C-terminal tail region. You can see where the flavin, the FAD, and their tryptophans are in the molecule. And apparently the C-terminal tail, based on some experiments, is really the only part that's ultimately needed for sensitivity to Earth's magnetic field, which raises the question of how this thing really works. If the rest of the molecule, including the flavin and the tryptophan, are gone, what has happened? So it seems to be a functional paradox that in Drosophila, fruit flies, for example, magnetosensitivity can depend only on the C-terminal tail, the CTT, without requiring the radical pair forming domains. Does it get the radical from somewhere else? I don't know. This remains a mystery. Additionally, avian studies have revealed that the light-activated steps are followed by light-independent magnetosensitive processing. And this challenges the simple short-lived radical pair models and suggests that more long-lived models may be needed in order to explain how magnetosensitivity can occur after a photon has been absorbed, but after the light has been removed and the magnetic sense is retained by the organism in the dark. And I have some papers you can read here relevant to these results. These are really the key papers. So, I mentioned earlier that cryptochrome and photolyase are very similar. In fact, they make up a superfamily of proteins with comparable photocycles and comparable electron tunneling pathways. This is an ancient flavin-based protein architecture, and the two proteins share very similar structures, similar protein folds, but usually diverse functions, although there has been found to be some overlap. I will say, for the most part, cryptochromes are blue light photoreceptors, and photolyase are blue light driven DNA repair enzymes. So cryptochromes typically do not have DNA repair activity, although some exceptions have been found. If we look at photolyase here, you can see it looks similar to cryptochrome, but where the C-terminal tail region should be, the C-terminal tail is missing, and that gives a spot for DNA to sit as well. Now, this is very interesting because the way that cryptochrome operates, it has a pair of photoreceptors as well as a whole arrangement of photoactive tryptophan residues in the protein, and it can absorb a photon of light. This creates an excited state, an excitonic state in the system, and now through the charge separation process, in fact, an electron can be essentially fired down an electron transfer chain into the DNA. And this electron gun, so to speak, is used to break up a DNA lesion, ultimately repairing the DNA. So blue light photoactivation occurs. There's an electron tunneling process through a superexchange pathway, accompanying protein conformational rearrangements. This is not a static process for the protein overall. Similar to rhodopsin, once you create an excited state, the whole protein can undergo a change in its shape, a conformational reorganization, we would say. And in this case, downstream repair. The DNA is repaired and the protein reorganizes in order to recapture the electron and return to its resting state. Now, I mentioned that the C-terminal tail in cryptochrome is, as the evidence says, the only critical part. Of course, if the C-terminal tail is there, there's no room for the DNA, but you might imagine the electron being fired into the C-terminal's tail instead, if the same photolyase-like motif and action mechanism is retained in cryptochrome. So this is really interesting. As I mentioned, some animal-like cryptochromes exhibit dual functionality. They can either operate as cryptochromes, blue light sensors, or DNA repair enzymes. And substantial magnetic field effects have also been found to coexist with rapid singlet-triplet dephasing, and this reveals the robustness of the system even to ambient noise. So to understand the photophysical foundations, there are still outstanding questions, particularly in vivo, in the living organisms themselves, and many issues remain unresolved. What are the radical or exciton identities in the molecule? What competing pathways are involved in the electron tunneling mechanism? What really is the source of the anisotropy? Is it a conventional radical pair mechanism, or is another radical pair-like mechanism involving other electronic dipole interactions or scavenging effects? Ultimately, how does the receptor architecture control the overall mechanism and ultimately the amplification scheme? We really need a mechanistic framework that fully explains how to incorporate or whether we can neglect all of hyperfine spin orbit, dipolar exchange, and even spin vibronic couplings to the nuclear motion in the environment. And this is needed to ultimately provide a clear foundation for magnetically influenced photoreactions. Now, ideally, we can take an integrative approach to understand these systems that would require ultra-fast spectroscopy and other ultra-fast experimental techniques, high-resolution structural biology, as I mentioned, open quantum systems modeling, and system-level physiological investigations in the operation of these senses in the organisms themselves. What future directions are there? What are the applications? Well, there's a lot. Of course, this is significant for circadian health research, and it may shed light on light therapy, phototherapy, disorders related to sleep disruptions like shift work disorders, and other areas of sleep medicine. Magnetobiology has other applications in terms of understanding and developing navigation systems, biomedical devices, and environmental technologies such as sensor systems that can be used to combine a magnetic map of the environment with other environmental measurements. And now, radio frequency and electromagnetic fields have been proposed to have important biomedical effects, although these are not fully understood. And so understanding these processes, the role of radical pair type processes that may enable magnetic fields, even weak magnetic fields, to influence physiology, are important to understand and ultimately establish robust safety standards, therapeutic applications, and exposure guidelines. Now, molecular spintronics could also emerge from this because as we understand the radical mechanisms, the spin-dependent mechanisms in the individual molecules, we could use these to develop quantum sensors, compasses, of course, perhaps quantum memory and other spintronic devices, and bio-inspired electronics, organic electronics. This electron transfer process, radical separation process, is significant for understanding how we can develop molecular wires and even hybrid silicon organic electronics and photonics. Now, all of this can converge as we consolidate research in quantum biology, biomedical technology, bioelectronics, and spintronics in order to create, potentially very advanced biotechnologies that can interface between conventional electronics and living systems themselves. So thank you for your attention. If you found this lecture interesting, don't hesitate to download your guide to quantum biology today from the archive link shown. It's available now for researchers, students, and curious minds who are ready to venture beyond classical biological explanations into the world of quantum biology. And if you have any further questions, don't hesitate to send your comments or queries to my email at the email address shown. This is my YouTube channel. Thanks again for watching and have a wonderful day.